we combine the special seminar of our department, immunology department and virology channel. Welcome participant online, and I think this is our all from Virology Associate Thailand. Today we have a special guest from Malaysia, Dr. Ramapraba Appanna. She is working on dengue, but she is viral immunologist, mainly focused on B cell. The topic that she will share with us today is beyond the fever, understanding the immune response in dengue infection. I think Prabha can tell us about herself a little bit. I would give the floor to Prabha, please.
Okay, can I start? Yeah, okay. Okay, good morning, everyone. So it's definitely a great pleasure to be here. And first of all, thank you to Dr. Cobb and the Department of Immunology, Faculty of Medicine, Sri Raj Hospital, Mahidol University, for giving me the opportunity to share with you my scientific experience, especially on dengue. And I think I'm truly proud to be here in this land who have made a significant contribution in understanding the tropical infectious disease, especially on dengue as well. So as mentioned by Dr. Cobb, so my title will be on Beyond the Fever, Dissecting Host Immune Mechanism in Dengue Infection. And majority of my data that I'm going to show it to you today will be from my postdoctoral training in Singapore. So okay. So so as you can see here, so I have completed my PhD in Malaysia, then on dengue and immunology. So basically, I graduated in viral as a viral immunologist, and then I moved to multiple different places across the globe for my postdoctoral training, starting from Singapore, where I continued my work on dengue and particularly on B cells and monoclonal antibody. And then I moved to Italy to study on allergy inflammation and on B cells. So this is work on a clinical translational on eosinophilic esophagitis, and then to the United States under the Gates Foundation. And this is working on H1N1 specifically on maternal immunity and vaccine. So as all of us know, I think it's very famous. Over a decade ago, when I started working on dengue, dengue always remains until today, I think, a huge public health concern. So you can see that how it is distributed across the globe, not only in Southeast Asia, but also in other continents of the world, including in Africa and also in South America, especially in Brazil. So where a majority of my study was conducted in dengue in Malaysia and also in Singapore at the time, I know that about 60% of the population, both asymptomatic and symptomatic individual have actually have seen dengue at least for the very first time in their life. So almost all of them, 60% of them already been infected. But what is good now is that the mortality with a lot of understanding which is made on dengue with a lot of important discovery on biomarkers and therapeutics, antibody, and eventually now we have quite a promising vaccine which is still under a lot of consideration of many countries. I think the mortality of the rate of dengue has significantly reduced. So I think just to recall a little bit of the memory of everyone, we know that dengue actually is an acute febrile illness. So majority of the children or adult who are infected with the virus can completely have asymptomatic clinical symptoms. So that means they don't show any kind of a symptoms, or they can have a very mild fever with a very specific clinical features here. And what is the most prominent clinical features, as all of you know, is the plasma leakage. This is the most severe form of the dengue shock syndrome. And then you also, as the leakage takes place, there will be a rise in the hematocrit, about 20%. And if it's not treated well, both in children and adult, this disease can progress to dengue shock syndrome. Eventually, if it's not well treated, that it can cause mortality. So I mentioned in my title, it's Beyond Fever, and it always been beyond fever, the understanding of dengue or the complexity of the disease itself. So as you can see here, the disease itself can be divided into 3 different phases. Febrile, both in DF and DHF. It's a febrile phase. You have a huge rise in the temperature, and then you have a defervescent phase. So this defervescent phase is a very critical phase where majority of the clinicians will take, when the fever drops, either the patient recovers completely, which is seen in dengue fever, or there is a high possibility, depending on the many defined factors for now, that the patient can go into a plasma leakage and progress into dengue hemorrhagic fever. And the prominent features here, as mentioned, they have a drastic drop in the platelet count, and there will be a rise in the hematocrit. If this is seen in the patients, then there is a high possibility this patient is going to progress into dengue hemorrhagic fever. So a lot of understanding so far has been made what is actually going on during the defervescent phase. And also definitely either the patient recovers, which always we hope at the convalescent phase. Unfortunately, some of them can also can go into a mortality at this phase. So there is a lot of defined immunopathogenesis of dengue. I think all of you in this room already know about this, but just to refresh the memory again, historically, the antibody-dependent enhancement, where we called it as ADE, is a defined factor of the pathogenesis of dengue, which has always been seen in both in children and also in adults. So this is a contribution of the non-neutralizing antibody which happens or being activated and secreted during the heterologous secondary infection, which could eventually enhance the viral uptake and replication via the Fc gamma receptor on the antigen-presenting cells, such as dendritic cells or monocytes. And then the second factor is the original antigenic scene. It's actually associated with both CD4 and CD8, and the understanding is actually very similar to ADE, where the immunopathogenesis takes place in a sequential infection with a different serotype, heterologous infection, and then you have a lot of clonal expansion of a low affinity TCR binding to the viral peptides and secreting a lot of non-specific cytokines, which actually delayed the viral clearance and also could potentially lead to the disease progression. And then, of course, the HLA itself. So HLA do plays a very important genetic factor that many studies has been done, including I myself during my PhD work, that human leukocyte antigen, both class 1 and class 2, can be a defined biomarker to say that whether these patients can actually progress to more severe or confer a protection. And this highly differ by the ethnicity and where does this patient is coming from geographically in a different population of the world. And then finally, the cytokine storm, which is a major contributing factor for the vascular endothelial damage and eventually the vascular leakage. And majority of these cytokines are associated with the inflammation. And then it actually could disrupt the tight junction protein of the endothelial cells. So this is actually historically a defined immunopathogenesis of dengue, which always taken into consideration when a dengue disease is being understood. So looking into dengue vaccine, there were 2 dengue vaccines so far has been kind of generated and being studied. And if you can see here, one is the Sanofi Pasteur and the other one is Takeda. So you can see the overall vaccine efficacy of Sanofi Pasteur, and we all know that this vaccine efficacy was relatively very low in seronegative participants. However, it becomes significantly higher in seropositive. That simply means that this vaccine highly dependent on the existing immune memory to activate the vaccine-specific antigens. And also WHO has strictly advised, and this vaccine should not be delivered in anyone or any individual in the endemic region because of the expansion or the generation on a low affinity antibody that can put the patient into risk. And this is again supporting theory of the ADE. So this vaccine has substantially not convinced by many people or many countries around the world. However, in the latest progress of the vaccine development, we have a Takeda vaccine, which is more promising and is taken into a lot of consideration by many countries, including Malaysia and Singapore as well. And then you can see here the vaccine efficacy for both seronegative and seropositive individual is almost similar. So that it looks like this vaccine could work better. However, if you look into the detail of the vaccine efficacy coming out from multiple different countries, shows the higher efficacy for dengue 1 and 2, but not observed for dengue 3. So there's a lot of limitation because of, I think the limiting factor here is the animal model itself. So how can we study or we gain a deep understanding of dengue if a lot of factor is actually missing to understand the disease? I think a lot of effort has been going on for 100 years almost now, but this is the best we have in our hands, and there is a lot of gaps to understand the disease, and this open up a lot of different kind of research questions. So going into a little bit more specific on how, for the rest of my talk, is going to touch on how the humoral immune response is working or has been working during the dengue infection. And as you can see here, the B cells will definitely get activated in a different form as the dengue virus antigen. For example, here dengue 2 engages with the BCR, then this particular B cells communicates with the T cells, and via the CD4 signaling from the T cells, these B cells can get activated and differentiated to antibody secreting cells. So the second signaling can occur when the dengue antigen bind to BCR and the viral nucleic protein bind on the TLRs. So this is the second way of activation of the B cells. So both this activation give differentiation of this B cell to antibody secreting cells. However, what happens during dengue is there are mixture of antibody is being made as the B cells start secreting the antibody. They can be both low and high affinity antibodies. And the high affinity antibodies making B cells are generally recruited to become a memory B cells. And this is generally a germinal central output. And the complexity of the disease, as I mentioned many times previously, occurs during a secondary heterologous infection when the patient is seeing the virus, different kind of a virus after a couple of months from the previous infection. For example, in this case for dengue 3. So the immune system remembers the infection by the virus rather than the serotype specific. So the B cell receptor remembers the dengue 3 infection now as a dengue infection. So it just activates the memory B cells from the previous infection, and they start generating almost completely a low affinity antibody. So this particular clearance of the virus are highly dependent on the avidity or rather than the affinity of the antibody. And this also masks the opportunity of the specific B cells of dengue 3 to get activated and to perform a high affinity secreting B cells. And yet, as you can see here, is a snapshot of what is going on during the infection. There are multiple different types of memory B cells can be made from the previous dengue 2 infection and coming out to dengue 3 infection. So what we observe, interestingly, during a secondary heterologous infection, so since the heterologous infection is causing the complexity of the disease, a lot of these research questions were actually taken over during or made during the secondary heterologous infection. And what is being seen is a strong plasmablast expansion during a secondary heterologous infection here in compared to the primary infection or other kind of febrile illness. If you see, it's a significantly higher at the very acute phase of the illness in the adult dengue patients. And if you look into other kind of viral infections such as flu, we also found a very specific feature of a strong expansion of a plasmablast. And this is actually true in the secondary heterologous infection and also a primary infection in dengue in comparison to flu or other febrile illness. So this looks like a very interesting feature for dengue. So the research question we ask here is if it is such the plasmablast is expanding so greatly during a primary and secondary infection, so what is the specificity of this plasmablast and how does this repertoire actually change during the infection, and also what is the function of this antibody which is made by this particular cells, plasmablast. So to address this in-depth question, we actually started the entire experiment just with 2 patients, as you can see here. So we recruited the patient at the very early stage of the infection, and this is the sequential of the infection. For the first patient, we had a previous of dengue 1, followed by dengue 2 infection, and the second patient at a previous of dengue 2, followed by dengue 3 infection. And then we recruited the patient at the very early and at a very acute phase of the disease. So this is where we actually pull out the antibody secreting cells or the precursor of the plasma cells, which is the plasmablasts. So the entire work and the understanding of the plasmablasts were done using a single cell technology. So we sorted out the plasmablast population into a 96-well plate, and then we amplified both the heavy chain and the light chain of the B cell receptor or the antibody for IgG and IgM. And then we took the pieces of the DNA here, we clone into a respective vector, and then we express it, the antibody, into a IgG1 expressing system. And then we studied the specificity of the antibody to understand where exactly they are binding on the virus. And then we did an in vitro neutralizing experiment assay to see also how much of this antibody are being protected inside the mice. And then to understand the genetic makeup, repertoire analysis, and how much affinity maturation have this B cell has achieved, we also made a sequence analysis on the B cell receptor, both IgG and IgM, including the Ig kappa. We started the entire experiment with the Ig kappa, excluding the Ig lambda in this research. So interestingly, what we found is about, so we made about, as you can see here, about 60 plus antibodies, and we found majority of this plasmablast antibody, which is made by this plasmablast, are dengue specific, you can see here. And the rest of it are actually a bystander activation, we would say, and they are not binding to any part of the virus. And surprisingly, though we have the information on the sequential infection, majority of this antibody are highly cross-reactive across all the 4 serotypes. This is what we saw. And interestingly, almost all of them, or 80%, at least above 80% of them were binding to the E glycoprotein, and they were binding to the they were actually binding to both the previous infection and also the current infection for the patient 1, and also the true thing was also seen for the patient 2. So they were highly specific to the E glycoprotein, these antibodies are. And in our mouse protection experiment, the interesting thing that we saw and which support our hypothesis is actually, so if you look at the how the behavior of this monoclonal antibody against the previous infection for both the patients in comparison to the current infection, we found that they were neutral in comparison to the control antibody here. Most of our monoclonal were neutralizing the serotype from a previous infection rather than the current infection. We also see in the current infection there is an enhancement rather than neutralizing factor. So this clearly support the hypothesis that what is being activated during the secondary heterologous infection is a memory B cells, which is seeing the virus as a virus itself rather than a serotype-specific activation. So we went about studying the genetic makeup of the dengue-specific monoclonal antibody, and what we found was is actually. So what we found is actually a majority of the dengue binding monoclonal antibody from plasmablast are having a very bias to VH1 gene usage for heavy chain. And also this is true for the light chain. And if you can see here, the non-specific memory B cells, that means we, along these plasmablasts, we also pulled all the memory B cells, and we found that majority of them are binding to the VH3. So that means there is a high potential that these dengue-specific antibodies are biased to a VH1 gene usage, at least for these two patients in our first study experiment. And out of this first 60 plus antibody that we made from these two patients, we were lucky enough to identify one highly potent neutralizing antibody, which were neutralizing all the four serotype at the nano to picomolar concentration. And as you can see already in the graph, the neutralizing ability is really good. And we also know that, interestingly, this particular, as mentioned earlier, the majority of them were using VH1 gene. And this highly potent neutralizing antibody were using VH146 and VK133 gene, and it has a remarkably high long CDR3 amino acids. So this gives the opportunity of the antibodies to stretch between the E domain 1, 2, and 3 to bind to the specific epitope that requires a good, highly potent neutralizing capacity. So in the first form of the study, we found that majority of the B cells are polyclonally activated. I think I'll show this slide in my next coming slides. And then they're highly cross-reactive across all the four serotypes, regardless what the infection sequential looks like. And they are binding to the envelope glycoprotein. For both on monomeric and dimeric structure, and they do have a weak to moderate neutralizing capacity and confer protection to the previous infection rather than the current infection. And one highly potent neutralizing antibody has been identified, which could neutralize across all the four serotypes. And interestingly, they belongs to the germline of VH146 and VK133. So the next question we ask obviously is what is the features of the memory B cells if we understand what is the feature of the plasmablast. One would assume that in the same patient, the genetic makeup of the plasmablast and the memory B cells can be very symmetric. However, let's go into the deep to understand what is going on in the patients. So as you can see here, the memory B cells, and we know that we in the blood, we have a lot of memory B cells against many kind of antigens that make immune surveillance to protect us. So how do we pull out the memory B cells against dengue is to create the right probe to bind on the B cell receptor, and then we actually fish out the memory B cell, which is specific for dengue, from the same patient as we pull out the plasmablast. So in the second study, we actually took, rather than the two patients, we actually took 12 patients both with the secondary infection, heterologous infection, and also patient which is seeing the virus for the very first time. And here we actually collected not only the plasmablast, but also the dengue-specific memory B cells. So we sorted out the cells at the different time point of the infection and, of course, after the disease recovery. So we did multiple different type of analysis here. So the serum analysis were made to understand the global immunoglobulin responses against dengue. And also we sorted out the plasmablast as the previous study and dengue-specific memory B cells. And we also performed A54 sequencing. At that time, about in 2016, this was one of the important next generation sequencing to understand what is the in-depth of the B cell receptor and the genetic changes of the B cells in the dengue infected patients. So as I mentioned earlier, how we sorted out is we used the exact city molecules to fish out the plasmablast, and also we have a probe. We created a probe for dengue 1, dengue 2, and dengue 3, and then we pull out the dengue-specific memory B cells as such. So as mentioned earlier, we found that the VH1 gene was particularly of interest for the plasmablast. And this is true as we expand to more patients in the second study. As you can see here, the expansion was seen for patient 1 and 2. And then as we analyze the memory B cells specific to dengue, you don't see these features anymore, or it has been shrinked. And then this is true also when we do a deep sequencing on the B cell receptor. And then, however, as we increase more of these patient numbers, we found that, the VH1 gene usage is not completely specific for dengue. So different patient could behave in a different way and use what is available in the repertoire to generate dengue-specific antibody. And this is true for plasmablast and also for memory B cells. So as I mentioned earlier, unlike other viral infection, sorry, can I have that removed? Because the title is blocking the, yeah. So as I mentioned earlier, there is a little clonal expansion of plasmablasts were seen in all the patients we have studied. So unlike other viral infection like flu or HIV, when a B cell is trying to make an antibody, you can see a huge clonal expansion. That means that the B cell receptor is going to use the exact genetic makeup of that particular specific high affinity antibody to generate more of these B cells or more of this antibody. But this is not actually the true what we found in dengue. And as you can see here, majority of this, they do have a little expansion. That means the CDR3 and the VDJ recombination is the same, exact same. And some of the CDR3 have gone through about 85% of a mutation to increase affinity maturation. And then some of them remains without mutation. That means the CDR3 is actually highly identical. And majority of them do only have individual, it's not even an expansion, individual activation. So this is the kind of a feature, except for the patient 6, so that we found how these B cells are being activated. So how do you actually fish out the most high affine antibody when the B cell is not informing us anything? So I think it's kind of difficult. This is one of the difficult thing also we went through, or challenges that we went through to identify the highly potent neutralizing antibody. Doesn't work. So here, what I made is like to understand what is going on in the plasmablast and the memory B cells. We made about more than 120 antibodies in total, monoclonal antibody from the same patients. And then we found that, as previously reported, we found that about 85% of this monoclonal antibody was still binding on the E-glycoprotein. So one would assume the behavior of these B cells on the monoclonal antibody, where exactly is binding in the humans, are actually going to be the same if you pull out the plasmablast and the memory B cells. But this is not the case for dengue. So what we found that, as I mentioned, here is the E-glycoprotein, but then if you pull out the memory B cells from the same patient, you found that the specificity changes were actually seen. As you can see here, now you start seeing that about 20% of them were actually binding to the for the immature virus particle PRM on the immature virus particles, and also about more than 50% were binding on the conformational structure of the virus, which is present both on the immature and the mature virus particles. So that means that the dengue-specific memory B cells do bind to more wider side of the virus rather than very stringent part behavior, which is shown by the plasmablasts. And then looking into very quickly, looking into how this B cells or this monoclonal antibody were neutralizing the antibody. Majority of this clone were actually neutralizing the recombinant E protein much better compared to the complex epitope. This is actually all the monoclonal antibody which was studied and derived from plasmablasts. And this is actually true also for the memory B cells. Though we found that some of the neutralizing capacity were higher for PRM, and we understood that this PRM is not specifically a unique of PRM, but do have a component of the E protein inside here. So we concluded that rather than a complex epitope, majority of our monoclonal antibody were neutralizing the recombinant E much better compared to the complex epitope, which requires more high affinity B cell receptor to bind on the conformational structure which present on the complex epitope of the virus. So our interest for this study was, since we found that the specificity of the B cell, the plasmablast and the memory B cell were actually different, we went about studying whether what is the genetic makeup of this particular B cell receptor and how they are actually identical or how different they are between the plasmablast and the memory B cells. As you can see here, the overlapping, there almost no overlapping in one of the patients between the plasmablast and the memory B cells. If you can see here and here in one of these secondary infected patients. And also, even there's a very, very small number of clones are being shared. And this is also true for the patient who going through a secondary infection and also primary infection. And interestingly, in this study, what we found was when we take a total of what is being shared between the plasmablast and the memory B cells, we pull out about 60 plus sequences and overlapping sequences, and then we overlap the plasmablast and the memory B cell here. Some of these sequences which expanded greatly in the memory B cells, we found that majority, all these sequences were actually IgM. I think this was one of the great understanding that we made at the time on what is the importance of the IgM memory B cells for dengue, where the classical understanding are normally the IgG memory B cells. So I think many reports or many manuscripts or research has come out after this understanding. The highly potent neutralizing antibody for flaviviruses or other kind of viruses could come out from, can be extracted not only from IgG memory B cells, but also from the IgM memory B cells, where they do play a prominent role in neutralizing the virus. And we also went about studying the clonal relationship. That means it's not only the identical clone, but a clone which have mutated, but they still shared the VDJ recombination. Yet when we analyzed the clones in this way, we found that they exact the same kind of a feature which we found before, where there is very little sharing between the plasmablast and memory B cells. And again, very interestingly, we found not only the importance of the IgG memory B cells, but also the IgM memory B cells for dengue, especially for dengue-specific memory B cells compared to the plasmablasts and compared to the dengue-negative memory B cells. So we concluded that in the second study that the memory B cells are highly cross-reactive, still highly cross-reactive across all the 4 serotypes. They do bind to a wider range of epitopes. So now they do bind to the more complex epitope, and also they bind to the pre-RM protein on the immature virus particles. They still have a weak to moderate neutralizing capacity. So it's very, very challenging to have one clone that or multiple clones that which has a feature of highly potent neutralizing capacity for dengue. And they neutralize much better the envelope glycoprotein. That means the affinity maturation seems to be very moderate rather than highly affine. And we found there is no clonal relationship between the plasmablast and the memory B cells. And interestingly, I think this is one of the beautiful understanding for this study is the importance of the IgM memory B cells, the role of this IgM feature in the memory compartment for dengue. So in the last study, which I was involved in as a postdoc for at least for the dengue projects, we use a combination of a single cell transcriptome and a BCR cloning approach in parallel and in combination to understand what is the specificity of the plasmablast. And while we took out the transcriptome, we also give the opportunity how these B cells could potentially being activated from the precursor of the cells and what could be the fate of these cells. So it's a very complex slide, I know, and bear with me that I will go through very quickly. And what we found was the plasmablast. So we actually kept the consistency of our plasmablasts across all the 3 studies, where we pulled out the CD27 high and CD38 high plasmablast cells rather than the plasma cells, which is the precursor of the plasma cells, which is being activated during the dengue infection. In the heterologous infection, they do have a mix of origin. So where do they come from, how they are being made, and where are they going next is being able to understand by the single cell transcriptome analysis. So interestingly, what we found in a very classical understanding, we always say that there is only one cells which is called as plasmablast. Yes, that's true, but in this study we found there is 3 different clusters that give us an information how this, which is we called it as cluster 0, 1, 2, and 3, and by analyzing how these B cells could have been activated and also clustering them together, we found that in the cluster 0, 1, we found there is majority of the cells do have a feature of the extrafollicular response. That means they do not need the help of the T cells to get activated, and they have the feature of the memory B cells. And in the cluster 2 and 3, they're highly dependent on the T cells, and they still do have the feature of the memory B cells, but then they don't have any feature of the extrafollicular response. And if you see the percentage of the IgG abundance, it is seen to be much higher in the extrafollicular response that give us information. Potentially, these cells are going to be recruited into becoming a long-lived plasma cells. And here they are lower, that probably these cells is going to remain as memory B cells to do immune surveillance for the next infection. So I kind of summarizes what is going on in this slide. And of course, I open up for questions and answers much later. So interestingly, in our previous study, we found that the VH146 in that 2 patients we study which gave us information of fishing out the highly potent neutralizing antibody. Interestingly, this was a very understanding that we found that majority of our clones, antibody clones, when we overlap with the gene, majority of these clones were using the VH169 here, as you can see. So the VH169 is a relatively very interesting germline gene where a majority of the antibody, highly potent neutralizing antibody, not only for dengue, but also for HIV and influenza virus has been extracted from this germline VH169. So that gives us an understanding that the B cells from cluster 1 probably going to give us much better opportunity to extract highly potent neutralizing antibody, which probably have the component of VH169. And also they do have a sharing of IgHJ6 gene in combination. So if you look into the highly potent neutralizing or the neutralizing capacity of this antibody, we found that there are a small subset of these antibodies from the cluster 01 had a better neutralizing capacity in comparison to the antibody which is coming out from cluster 2 and 3. So in this very last study, I think this is my last slide on dissecting on the humoral immune response for dengue. I think we kind of concluded that in cluster 01, majority of the cells do not require a T cell help. They are extrafollicular response, and probably they do not go into a the germinal center interactions. And they do have a feature of a long-lived plasma cells. That means this could potentially help in the antibody levels inside the blood after the disease recovery. They do have ability of much better neutralizing antibody pool inside here, and they do have a specificity of binding to complex epitope and non-fusion loop, and they have a shared VH169 gene. And in cluster 2 and 3, they are T cell dependent. So these B cells are still going on with the affinity maturation process. They do have a feature of the memory B cells. They still remains as a low affin, and they are specific to fusion loop. So as you can see here, I kind of took all the information and made the graph. What does this combination of a single cell transcriptomic and the BCR cloning approach could finally give us? So it gave us a great understanding of the origin of the cells and how these cells could have been activated to become a plasmablast during dengue infection. So it can be through extrafollicular response or a T cell dependent responses, and what is the fate of these cells, what potentially these cells were becoming. So from the cluster 2 I mentioned, they have an opportunity to become a memory B cells, and then from cluster 01 they have a great opportunity to become a long-lived plasma cells. So with that, I think that particular work will not be possible without all these great people I have worked with in my scientific journey, especially the members in Dr. Fing's lab in Singapore Immunology Network. And of course, it was done with a great collaboration with the bioinformaticians and immunogenetic platform at the time in Singapore, and also for the rest of the scientific team in Italy and also United States, and all the grant funders for this project. Thank you very much for your attention.
Thank you, Prabha. Any question from here? Let me warm up with one very simple question. Since we have a lot of students here, right? Seem like at the beginning you're talking about the technique that you look at B cell based on single cell sorting, right? And later on you kind of like introduce single cell transcriptomic. Can you give us kind of like advantage, disadvantage? What would be the best way to dissecting B cell response in viral infection?
Sure. I think this is a very, very important question. It depends how deep do you want to understand the behavior or the characteristic of the B cells. So if you use a single cell sorting, I think I prefer to go back to my methodology slide.
Which slide?
The methodology slide.
Number.
I can't remember the number, but this one, the 11, please. Perfect. So one more click. Oh, can I click here? Oh, okay, perfect. So in this particular study, as I mentioned earlier, it depends on how deep do you want to understand. So this particular methodology stopped at the understanding of the B cell receptor, gave an opportunity of where exactly the B cell binding, because when you do the monoclonal antibody, you're going to understand where exactly they are binding. And then they also, when you sequence the B cell receptor here, you're going to give us an understanding what is the repertoire, how much the B cells have affinity matured. But then when you do a 10x genomic, it's going to give us how these B cells are getting activated. What is the pathway has been used for the B cells to get activated? And what is the immune programming of these B cells? So that information will be missed in this methodology. So this methodology just stopped at the, as I mentioned earlier, what is the repertoire, what is the affinity maturation, where exactly they are binding by cloning the B cell receptor. Yeah.
Okay. Talking about like the yield that you get specific B cell to produce the monoclonal antibody, which method would be better?
Okay, so I have to say that when you see here, or at least I go back to the work that I have done by myself. It's this one. So this is entirely the experiment that I've done. So you see here, in the plasma blast, you are not creating any kind of a probe here. So what you're sorting out is completely a plasma blast population. That means that you are using this particular marker and then you're sorting out the plasma blast, right? But then for the memory B cells, we are creating a probe here. So that probe of the virus particle is going to bind on the B cell receptor, and then you're fishing out the memory B cell, which is specific for dengue. So there is a slight bias here, and there is no bias here. So going back to your question, so here is exactly what is happening in the human body, right? So you see here about 85% of them, at least 75. In the first experiment, we know that about more than 70% do bind on for dengue. So the rest 30% is just a bystander activation, because bystander activation happens because there are a lot of cytokines are being made during the dengue infections. The B cells can not specifically get activated. But then here we know that 85% bind to the E glycoprotein. Unlike the memory B cells, there is a slight bias, but we hope that this is the best we can produce of what is going on in the human body. Yeah. Did I answer your question? Okay.
Hi, great talk. Referring back to the single cell analysis, you mentioned that you perform both SmartSeq and 10x genomics. Just wondering what the differences are and what advantages each of the technologies would make.
That's a very great question. So I have to go back to the. So the entire study, while she set up, so the entire study, the 10x genomic, is the down one. This one, yes, yeah, this combination, 27. So the entire study started with a SmartSeq first, okay, at the time. So, and also, then it continued with 10x. I think the SmartSeq and 10x depends on the depth of the sequencing, how much sequence you're going to get from the SmartSeq and also the 10x and also the length. So both has the pro and the cons. And both is going to give us an opportunity to analyze the data. Because when we combine the SmartSeq and also the 10x here, the important things the reviewer were asking us, do you see difference? Actually, we were working very closely with the bioinformatician to tell that there is no difference that we have seen between the SmartSeq and 10x in terms of the regulation of the gene and when we did the pathway analysis. I think one is how much deep you can sequence it, and then one is how much the length of the sequence, the 10x genomic. Yeah, I think the 10x, the SmartSeq were used to, what I did was when the SmartSeq was performed, I used the pieces of the DNA there to make the antibodies, not from the 10x genomic. So I think if I address your question correctly, I think it also it about the depth of the sequence that you're going to pull out, yes, and the length.
Okay, thank you. So in this figure that you had here, is this an integration of the two technologies?
Yes, it is. Yeah. As you can see here, the A is sorry. Here is the SmartSeq, and here is 10x genomics. This is because the reviewer were asking the combination of this technology, because at that time when we published this work or when this work was being carried out, the world was establishing this technique, so it was important, though we started with SmartSeq, it was important for us to combine the 10x. And you see here the plasma blast was taken, but then here is the entire B cells were taken. So that means that whatever you are seeing here should overlap on this entire B cells. Did I get you?
Yeah, thank you. If not, I have another question. Okay, I have another question. So in your single cell analysis again, you had your 3 clusters, cluster 1 and cluster 2 and 3.
Yeah.
I can see that there is differences in between 1 and 2 and 3, but I was wondering what are the key factors that deviate cluster 2 and 3?
That's a very important question. Actually, the cluster 2 and 3, if you can see here, the percentage of the specific IgG, which is very low, and then the metabolic activity, actually we can't really significantly, how do I say, we can't really significantly, immunologically, we cannot really define them. So we try to pull out, because when we do a gene analysis, it have to give a logical sense of, logical immunological sense associated to the B cells. So why we combine the cluster 2 and 3 together is because immunologically, both this cluster 2 and 3, though they look different here, as you mentioned, if you ask me what are the genes associated, which is differing them, I think I will not able to answer you now, but what is the gene which is putting them together are the immunological gene, which the behavior of the cells are dependent highly on T cells. So I think when you do a single cell analysis, I think you have a research question first. And then you see how is the behavior of the cells based on your hypothesis rather than acting on the gene itself. I think you need to have a hypothesis of this is what is my hypothesis, and you go about analyzing the gene rather than taking what the information the single cell analysis is giving. Yes, you take the overall information like here, cluster 01 and 02 and 03, but then if the gene information of cluster 2 and 3, that the differs between cluster 2 and 3 is mainly here is the surface markers. So we try to kind of do a wet lab experiment to prove that there is a difference between cluster 2 and 3, and it was very, very challenging because these clones were behaving in a very different way eventually when we extract the cells. So I have to say that I have to be very honest with you that first of all, you have a research question, and then you go about analyzing the gene. Of course, you are not going to create a bias there because what is given to you is exactly what is available there. It's just that you have to go about with your hypothesis rather than you act on what is being defined in the single cell analysis. Yeah.
Thank you.
Yeah.
Thank you so much.
Yeah.
All right, great talk, very great talk. So since you're on this slide, so I might have missed it. So how many samples that you use for the 10x? So that's
There's only
5 for smart seq.
yeah, and there's a 2, yeah.
So do you see the heterogeneity in this result in terms of like cluster 1, 2, and 3? So
No.
they are the same.
No,
then-
because we try to put the clinical features of the patient as close as possible. The patients were not very much on the sequential infection. So when the patient has dengue 1 and dengue 2, we cannot collect all the 5 patients which had the dengue 1 and dengue 2, but then we made it as a heterologous infection. So if you see here the patient who are in red, they all have a heterologous infection, and the behavior of the cells is the same. So if it's a primary infection, then it's going to be different. But if it's a secondary infection, regardless of what is the sequential of the infection, it's going to be the same. Yeah.
So they all have like cluster 1, 2, and
Yes.
And then-
Analyze that individually. Yes.
then for the next slide that you mentioned about the IgHV69, is that like patient specific or on the next slide? Yeah.
The next slide.
Yeah. On that 69 that
Oh,
turned red.
no, it's a combination. So what I made, so when the cells were sorted, so I took the pieces of DNA and then the monoclonals were produced. So the monoclonals that you see here, these clones here, is a combination of all the patients. So this is actually how do I say? It's a mean of all the patients. Yeah, VH169. Yeah, it's not one patient, it's all the patients. Yeah.
And another question that I'm pretty kind of new to the dengue field. So when you
Of
talk
course.
about the little expansion of the
Clonal expansion.
clone expansion,
I just
so any hypothesis on that, and then whether I believe that most of the data come from the adult, right? Is it the same situation in the kids and then in the elderly?
Yeah, let's go back to the slide that everybody understands here. The clonal expansion. I can't remember the slide number, sorry. I think up. It's the circle with the yes, it's five times. Up, yes.
Yeah.
This one, right?
Yeah.
So So this is actually a very important and a very beautiful question, I think, because this little clonal expansion. I have to tell you, to be honest, that it's very specific to dengue.
Yeah.
So, and it's not true for influenza or HIV that we always say that give us the HIV scientists and the influenza scientists a much greater opportunity to extract the highly potent neutralizing antibody, because the immune system is giving them the information that when I expand largely, that means I'm making a good antibody. But in dengue, the immune system is not giving any form of information. So this little expansion is going back to evolutionary theory, I think personally, because, dengue has been there for, I don't know, millions of years. I think it's just defined 100 years ago. I think it's same goes to the human B cell receptor. So I think they have kind of like this dengue, it's being recognized probably to the most innate like B cell receptor on the B cells. So that kind of like don't give much opportunity for the B cells to affinity mature completely and to expand greatly. I think because of the existence of the virus and also the immune system for such a long time, I think somehow they evolutionized to recognize the epitope on the virus. Many of these receptor in that repertoire is just recognizing the epitope of the virus. Unless for HIV, I'm not greatly sure how many thousands of years this virus has been there, but I think it give a very specific recognition. When the recognition is specific, I think the immune system have the opportunity to recognize, to decide, to affinity mature, and to expand. I think that is not taking place for dengue, because for dengue, the monoclonal antibody coming from multiple different repertoires. So that means that all the repertoires, majority of the repertoire in the human body are recognizing on the virus. So and the binding, I think it's not completely highly affine. I think it's a little bit loose. It's a little bit low affine and moderate. I think that kind of, but it still kind of blocks with the avidity and also neutralize to a certain extent. So there is no much opportunity for the B cell to expand, because expansion takes place because the brain friends to help them to neutralize. But if the B cell have many friends to neutralize the virus, the B cells don't need to use so much energy to expand. Did I get you?
I know.
You
But you think it's the same as if in the kids and then gonna be the same, right?
back in the kids, I think, I never did an experiment on the kids by myself. I think this is going to be the feature for dengue, regardless of kids. I think kids is going to be more because maybe in kids you might see, because the kids are seeing the virus for the first time and they've never been in this world for such a long time, unlike us, the adult. Okay.
Okay.
I think 1 hour go like very quick. And I think, by the way, for the people in the online, Prabha will be with us. Okay, one more question from student Ray. Come on here.
Sorry. Great talk, by the way.
Thank you.
And I have a few questions. I don't know if this might be too basic or not, but I was confused why you only experimented, or you only looked at the IG kappa and not the lambda.
No. We looked at both
You
actually.
looked at both.
No, for the very first experiment, you are right. We started the first. It was more technical. It was the primers that we had at the time for IG kappa. So the first study we only did for IG kappa, but then for the second study and the sequential study after that, we had for kappa and lambda. Yes. Yeah.
And could you elaborate on the results?
The results, if we see difference between IG kappa and lambda, to be honest, we didn't go back. I myself, when I started doing the lambda work, the lambda chain was very interesting, even though the lambda chain percentage is much lower in the human body than kappa. But I think the lambda chain was giving us much better specificity, also in terms of the percentage. And to be honest, we didn't really go about and looking into the details of the kappa and the lambda chain. But I was remembering that they do give a much interesting feature, the lambda, yeah, compared to the kappa, yes. Yeah, but honestly, I didn't go in depth to understand both kappa and lambda, the behavior.
Okay. I have one more question. Since you mentioned that there was a difference between the extrafollicular plasmablasts and the
T-cell
T-cell-dependent plasmablasts, and you mentioned that the extrafollicular plasmablasts produce higher affinity and more specific antibodies to dengue, then maybe could we produce vaccines or therapies to target these rather than the T-cell dependent plasmablasts, so that we can get specific high affinity clones and so that there wouldn't be as much cross reactivity and chances of like enhancement.
But you know, the understanding of extrafollicular response is it's beyond extrafollicular response, because I think the B cells, it depends on what is the arrangement of the amino acid on the B cell receptor, first of all, and how this amino acid is actually binding on the epitope of the virus. And then I believe that the extrafollicular response, the features, can we go back to the slide, please? I think it's better to. It's a very important question. There's the transcriptomic slide, 27. I remember the slide number 27, yeah. Yeah. Yeah, you see this, the extrafollicular response that we concluded here. First of all the patients here, majority of our patients here are from secondary infection, right? And I would assume, first, as I mentioned earlier, it depends on the arrangement of the amino acid on the B cell receptor. Even though it's a naive B cells, how this arrangement is made is extremely important, where they are binding on the dengue. Extrafollicular response, they do not need a help of a T cells. But I believe when a B cell already seen previously, already met with previously with the T cells, have already achieved sufficient affinity maturation. So when the B cells gone into the memory compartment, when they reactivate it again, if they have enough good arrangement and good enough affinity maturation, then there is no requirement for the B cells to meet the T cells anymore. Now that falls into extrafollicular response. That's why I say the extrafollicular response here is beyond the word extrafollicular response. Do you get it? I can talk to you later.
Okay.
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 You know, it depends on whether the B cell already met with the T cells. It's not, for example, in a primary infection, just to explain a little better, in a primary infection, if you tell me that the B cell is going through extrafollicular response, I don't think so that it's a valid thing that the B cell is going to make a good quality antibodies. Yeah.
Okay. Okay. Thank you.
All right. Thank you very much, Prabha.
Thank you, Kop.
Ray, by the way, Prabha will be with us for another 2 days. You can have a lot of time talking with her, okay? And for people who join us online, if you want to discuss more about B-cell immunology in infectious diseases, I think Prabha also has the experience with flu. You can come to Department of Immunology and go find me. Try to get me and I will introduce you to Prabha. Anyway, I would like to close the section today, and next time we will try to get more interesting talk for Virology Channel. Thank you very much.
Thank you. Thank you so much.